The topic of the lecture is causative agent of mycosis. Mycosis are diseases caused by microscopical fungi. Microscopical fungi belong to Eumycota kingdom. In past, the name of kingdom was Mycota, and so disease caused by microscopical fungi are named mycosis. Now classification is changed, and classification of microbes is revised very often. So now the name of kingdom is Eumycota. Anyway, diseases caused by fungi are named mycosis. Classification of mycosis in general. What diseases are caused by microscopical fungi? We are not talking about mushrooms. They cause food poisonings. We are talking about microscopical fungi. And this is classification of mycosis, disease caused by them. Systemic mycosis, and in some countries they are named systemic mycosis. At another countries, deep mycosis, like in Russia, they are named deep mycosis, and maybe in another country they are named endemic mycosis. So 3 synonyms are used. Had you tried all of them, you should realize that which one is used in your country. So by the name of diseases, it's clear that in this case, fungi are able to multiply inside the body, and many internal organs are involved, and it leads to lethal outcome. So the next group is subcutaneous mycosis, when fungi are able to multiply under the skin. The spores of fungi enter through the skin through some trauma, and in this case, fungi multiply just under the skin into the muscle. They go deeply and deeply from the place of entry, and inflammation with pus is developed under the skin, then inside the muscles till the bones. Sometimes ulcer is developed, and with pus, of course, bacteria are very often involved and cause secondary bacterial infections. Very often in countries with hot climate, this kind of mycosis. In countries with hot climates, people go barefoot, so very often they get some trauma by branches of trees, on arms, or on legs, due to contact with some bushes or branches of trees. Those are on the soil, and they are often in that countries. Then cutaneous mycosis, they are named dermatophytosis, dermatophytosis, epidermophytosis again. In this case, fungi are able to multiply in the skin, nails, and hair. They are very common in all countries. Then superficial mycosis, caused by microscopical fungi. Those multiply just in upper layer of the skin, epidermis, and some parts of the skin have another color, maybe a hyperpigmentation, maybe dark color, maybe white color. So it's not, they are not dangerous, but sometimes skin is not looking nice. Okay, so opportunistic mycosis. Opportunistic mycosis, like all opportunistic infectious diseases, are caused by conditional pathogenic species of fungi. Some species of fungi, so most of species of fungi mostly exist in the soil. There are favorable conditions for them. They are saprophytes, and they don't need a human's body or animal body, and they enter humans or animals' body occasionally. Nevertheless, they are able to multiply in human's body or animal's body. And some species of fungi, just a few, sometimes are detected in healthy people without any clinical symptoms. And you know that some of such microbes are named normal microbial flora. They are normal microbial flora. They are able to exist in 13 amount just in organs and tissues. Those are connected with environment. Of course, they are not, skin is not sterile, for example. Digestive tract is not sterile. So, and of course, there are microbes. Those are named normal microbial flora, but they should exist. The multiplication of them is suppressed by immune system, of course. They don't multiply as they wish, of course, and a microbes, those exist in 13 organs, have very complicated relationships in between. So some of them, like bacteria, suppress the multiplication of fungi, for example. And another bacteria produce bacteriosigns. Those are able to inhibit the multiplication of another species. So they are able to exist just at 13 organs, at 13 amounts, no more. They develop microbiocenosis in 13 in the nose, on the skin, in oral cavity, on mucosal membrane, in stomach. Now very modern to study this microbiocenosis, biotops. And what fungi are able to exist like normal microbial flora in very small amount? Candida, fungi of Candida genus, fungi of Penicillium genus are detected sometimes on the skin, and fungi of Aspergillus genus are detected sometimes. You know that the term microbial flora is an average. It doesn't mean that everyone has Candida or Penicillium on the skin. No, not at all. Just few people, a small amount of people have Candida, for example, on mucosa of mouth or mucosa of genital tract, maybe on the skin. So, and they are not able to multiply because of multiplication of them is suppressed by immune system and other microbes. But if they have opportunity to multiply, for example, on the background, any kind of immunodeficiency, they will multiply immediately and they will cause endogenic infections. Endogenic infections without any contamination from outside. So, and such infectious diseases caused by normal microbial flora are named opportunistic diseases, opportunistic infections from the word opportunity. And today we are talking about opportunistic mycosis. And one group is named mycotoxicosis. We are not going to study mycotoxicosis, but some species of fungi are able to produce very severe biological toxins. For example, fungi of Aspergillus genus. They are able to multiply on the food molds. Have you ever seen the growth of molds on vegetables, on the bread? They're very common because of spores of fungi. Really, molds exist in the soil, but spores of them are transmitted by dust particles, and they are able, if they will find some substrate that is available for growth, they will multiply. So on vegetables, on fruits, on bread, on some food, they are able to multiply. And if the food is infected by Aspergillus fungi, exactly species is named Aspergillus flavus, these fungi are able to produce exotoxin, and by eating all this food, person will develop mycotoxicosis, very severe sometimes, and this toxin produced by Aspergillus flavus is named aflatoxin. Aspergillus, in short, just A, Aspergillus flavus aflatoxin. And if it is accumulated in the food, very severe intoxications are developed. And even this exotoxin, aflatoxin, is able to produce malignant transformation of cells. So it is dangerous, and it is better to just throw away this food if there are some growth of molds. So just realize that there are a lot of mycoses, and some of them are very serious. By your program, we will study just this group, cutaneous mycosis, dermatophytosis, and from opportunistic mycosis, you will study candidiasis. They are very common, so you should know them. And I am going to remind you of fungi morphology. What are they? Fungi are eukaryotic cells. They have cell structure, of course, and they are eukaryotic cells. So they have all membrane organized structures. including nucleic membrane. So they have real nucleus, they have ribosomes, mitochondria, Golgi complex, and in cytoplasmic membrane, they have sterols. They belong to plants kingdom, so they have ergosterols in cytoplasmic membrane. And by the way, ergosterols are target of action for antifungi antibiotics, amphotericin B and nystatin. And the cell wall consists of chitin mainly, so it is different from the content of bacterial cell. And fungi are able to exist, fungi cells are able to exist in two shapes. Spherical shape. In spherical shape, always have Baker yeasts. Baker yeasts, they belong to fungi kingdom, and they have spherical shape. So, and if causative agents of mycosis have the same shape, they are named yeast like fungi. And for example, exactly Candida genus fungi, they have spherical shape, yeast like fungi. And molds exist in the soil. They have another shape, not so regular, just like branches of trees. This is one cell with many branches, and this one cell is named hypha. So molds exist in the soil like hypha, and if they grow on artificial media at room temperature, they grow like molds, and they develop such form hypha. Not possible to see, of course. We're talking about mycosis, and in diagnosis of mycosis, microscopical method is used, of course. Morphological properties are very important for identification. And if we are going to do microscopy of yeast-like fungi, we will see cells separately, one by one. And but if we will do microscopy of molds, not possible to see separately these hyphas. We will see aggregation of hyphas. And aggregation of hyphas is named mycelium. So this shape is named mycelial form, molds fungi. And these fungi in this form, they are able to multiply by budding, by binary division. Sometimes blastospores are formed by them, and molds multiply by spores formations. Exospores and endospores are produced by them, and they multiply by spores. So, and spores are inhalated by people with particles of dust. Not all of them. Just causative agent of systemic mycosis. Some species are dimorphic fungi. They exist in the soil like molds, like in mycelial form. But if they multiply in human body, they develop yeast form cells. And in laboratory, if they are cultivated at room temperature, they again exist in mycelial form. But if they are cultivated at the temperature of human body at thermostat, they develop yeast form cells. So not all species, just some of them. Now we will talk about cutaneous mycosis, dermatophytosis. Dermatophytosis, they are able to cause disease of skin, nails, and hair. Three groups of pathogens. Where do they exist in nature? Geophilic dermatophytes, zoophilic dermatophytes, and anthropophilic dermatophytes are distinguished. So, geophilic dermatophytes inhabit the soil, fungi of the genus Microsporum. Infection occurs after contact with the ground, with the soil, of course. The second, zoophilic dermatophytes, parasites of animals, cats, dogs, horses, cats, poultry, and so rodents, farm animals, and fungi of the genus Microsporum and of the genus Trichophyton. It is Latin, Trichophyton, not English. Infection occurs by contact with animals. Very often, children are infected by playing with these pets, dogs, and cats. And then anthropophilic dermatophytosis, human parasites. They are circulated among human beings. Fungi of the genera Epidermophyton, Microsporum, Trichophyton. Did you notice that there are three fungi of three genera are able to cause dermatophytosis? Trichophyton, Microsporum, Epidermophyton. Infection occurs through the household contact in baths, pools, swimming pools, showers, the common rooms for showers, through the water, as well as through sharing of combs, towels, hats, and shoes. If nails are infected, for example, it is supposed to be infected by sharing of shoes. If hair are infected, it is supposed to be infected by sharing combs and hats, and so on. Water, like vector, spores of fungi are transmitted by water from the patient to the healthy skin, and on wet skin, spores are able to multiply very quickly. People very easy are infected at these places, and then it takes many years to eliminate fungi. Fungi, unfortunately, are very resistant to antifungal preparations. So clinical aspects, what clinical forms now are described officially, maybe not important for you really. So tinea barbers each involve the bearded areas of the face and neck. Then tinea corporis is ringworm of the smooth of non-hairy skin of the body. Tinea capitis is ringworm of the scalp. No, maybe you will hear then. Tinea pedis, athlete's foot, is ringworm of the foot. Tinea manuum involves the hand, and Tinea unguium involves the nails. And clinical features. Lesions in the skin tend to be circular, dry, scaly, and itchy, and there is photo. Skin mycosis, multiplication of fungi in the skin, how it is looking. Then mycosis of the foot, multiplication of fungi on the foot, athlete's foot. Then nails mycosis, named onychomycosis, nails mycosis. Nails infected by dermatophytes are deformed, friable, and discolored, and finally they are destroyed completely. Have you ever seen such nails sometimes there? And they are destroyed maybe. So fungi really have no virulence factors of pathogenicity. But these fungi, exactly, they are able to produce enzymes for destruction of keratin, and they are able to destroy nails and hair. And they are able to multiply not just in the nail and in the tissue surrounding the nail. So it is difficult to eliminate these fungi. Then hair mycosis, FAWS, a chronic type ringworm in which dense crust develop in the hair follicles, leading to alopecia and scarring, so absence of the hair. There are photos of the patients with hair mycosis, and due to multiplication of fungi, finally, at first fungi multiply in one hair, and then it is destroyed. The fungi move to another hair, and it becomes dry, white, and then it is destroyed completely. Fungi move to another hair, and so on. They spread, and they are able to infect many hair, and multiplication of fungi leads to destruction of the hair. Finally, they are destroyed, and this is awful picture really of the patient without treatment. So microbiological diagnosis of disease of this kind of mycosis. Microbiological diagnosis. So what material should be examined? What specimen? Affected hair, destroyed hairs, some particles of them, then particles of nails and skin flakes from the place where the fungi multiply. And what methods are used? Of course, microscopical method is used. And firstly, unstained preparations are used, treated with alkaline because of due to and hot alkaline really, and the purpose is to destroy human being cells, and the fungi will be visible because some of them are inside the hair, some of them develop spores out of hair, and it's very important for identification. As usual in microbiology, identification should be done, and fungi have no so many property, but they have very specific morphology and location in the hair. And every sign is important for identification. The character of location of spores in the hair may be singly arranged, may be located in lines, and ectotrix out of the hair, endotrix inside the hair. So every this sign is described, and then it is possible to stain with methylene blue, hematoxylin, and eosin, and then again describe morphology. And also immunofluorescent test is used by using of specific antibodies. Then mycological method is used. Mycological method, cultural method is used. A specimen is inoculated on saburo medium. Saburo medium, very often used for cultivation of fungi. And these petri dishes are incubated at the thermostat from 1 week till 3 weeks, sometimes at 25 degrees by Celsius. It's thermostat at a temperature 37 and at room temperature 25 degrees by Celsius. Fungi develop colonies, and the colonies are R-type. Have you ever seen the molds grow? How do they grow? So they are whitish, cream, brown, red. They have some color, and they are looking like wool particles, like cotton particles, and so on. Colonies are different. Again, we should describe what colonies are developed by fungi. For example, Trichophyton rubrum. Rubrum is red. It's developed colonies with red color. Even the medium has red color. And now then we should prepare smear, and we should describe morphology of these fungi. What are they? Trichophyton rubrum. Then this is a colony of the fungi of Microsporum genus, Microsporum canis. They develop yellow color colonies, fluffy, and they have very specific morphology, bulged cells, and it's important. Sometimes really it is necessary to study biochemical properties, saccharolytic enzymes production for identification, but not very often. Sometimes it's enough to describe the appearance of colony and morphology. Epidermophyton flacosum morphology. Do you see the difference? So it is possible to identify by morphology and cultural properties. Then biological method is used. Inoculation, it is used really. Sometimes it is necessary to inoculate in the animals into the nail, inoculation into the nail, or inoculation to the hair for cultivation of fungi. And then again, the pure culture should be isolated, and we should identify fungi by morphology. Serological method is used for the detection of antibodies by using of agglutination test and ELISA test. And the main virulence factor of fungi, I told you they don't produce exotoxins, they have no endotoxins, but they are able substances of fungi are recognized by immune system like allergens, and allergic reactions are developed as usual. All microbes, some bacteria, some protozoa, but no viruses. Now we are talking about fungi. They are able to cause delayed type hypersensitivity, delayed type. If you study physiology, pathophysiology, you know that there are two types of allergic reactions. Immediate type reactions where immunoglobulins E are participated, but microbes cause delayed type of hypersensitivity, cell mediated. Special T lymphocytes after this allergen enter first time into human body. This allergen is memorized by T lymphocytes, and then if no allergic reaction is developed first time, but if this substance again enter human body, doesn't matter bacterial allergen, like in case of tuberculosis, leprosy, anthrax, brucellosis, tularemia. In case of protozoa like toxoplasma, like leishmania, if fungi, if they enter again human body, in the place of injection, there are special allergens. They are commercial, they are produced, and if in case of mycosis, very often skin allergic test is used. So if we know that positive agent, no, any species of microbes is able to cause allergic reaction, delayed type of hypersensitivity, we are going to detect this allergic reaction by using of skin allergic test. And allergens are produced. They are produced, and every allergen is named after disease's name, and it is consists of specific certain substances of the causative agent. Like in case of tuberculosis, it is a protein, tuberculin. Nowadays, it is purified protein derivate, just protein of tuberculosis bacteria is used for injection. And in case of anthraxine, just spores of Bacillus anthracis are used, and so on, some substances. So substances of fungi are used for detection of skin allergic for this delayed hypersensitivity. Treatment. So what about treatment? At first, treatment is done locally. Application of solutions of cream like clotrimazole. Water solution of clotrimazole is useful for onychomycosis, for treatment of mycosis of nails, water solution, if just at initial stage. Then creams and ointment like Lamizil, Nizoral, Mucospor. But sometimes if patient did not use immediately these medicines, they are not useful. And sometimes it is the with onychomycosis, griseofulvin, Nizoral, Diflucan, and sometimes it is necessary to take per os. These preparations, because of local application, is not useful. But unfortunately, these medicines that are used for treatment of mycosis are very, very toxic. And human being cells are eukaryotic cells, the same like fungi cells are eukaryotic. And in human being cell, there are the same targets for action of them. And unfortunately, we have very often they have a lot of side effects and destruction of many organs. Finally, it leads to dysfunction of organs due to treatment. Really, pharmaceutical faculty students, you know, every medicine has some toxic properties, every medicine, and develops different complications, okay? Even vitamins, if the person will take them in high doses. So, and sometimes it is necessary to remove the nail plate. The patient needs surgery, or there are pictures. If 6% of nail is destroyed, it's possible to use just local application, 5%. If 11% of nail are destroyed, necessarily to drink per os preparations. If 28% of nail are destroyed, the patient should take per os preparations for a long time. If more than 24% of nail are destroyed and maybe more, it is necessarily to take medicines per os and to do surgery to remove the nail. It's possible to remove the nail plate. It's not a big deal. And then we have to clean this tissue and place fungicides, again, medicines against fungi on the tissue, and the new nail plate will be developed. But anyway, fungi are able to survive in tissues around, and when a new nail plate will grow up, they will again infect this nail. So it's very, very difficult procedure with dermatophytosis of the scalp. Fluconazole is used, itraconazole, griseofulvin, terbinafine medicines, and there are pictures of them. Lamizil, then Lamizil cream, Lamizil spray, like aerosol to place on the skin, for example, what cream, and then flucarcin. Flucarcin is used antiseptic, and it is solution and we have to place on the skin and superficial wounds, erosion, and skin abrasions, flucarcin, to avoid fungi multiplication. Prophylaxis. Non-specific prophylaxis. Vaccine is not developed, of course, and just observance of hygiene rules. Skin hygiene using only personal shoes, hats, hairbrushes, avoiding contact with the sick animals, protecting the skin of hands from contact with the soil. So work in gloves. If you are working in the soil, be in gloves and don't go barefoot because of due to contact of soil, it is possible to be infected by fungi. Now I will make a break and then we will continue. In 10 minutes, we will talk about causative agent of candidiasis. The leaders, I will send you the link again in 10 minutes.
Okay, well understood. Thank you.